Hi, everyone. My name is Deema. I am a graduating biomedical engineer, and I had the privilege of working with Draper Laboratories to design a uterus-on-a-chip for the interrogation of trophoblast behavior in preeclampsia. So let's break that down really quick. The clinical problem we decided to tackle was preeclampsia. Preeclampsia is a pregnancy-related disorder, and it's one of the primary causes of maternal morbidity in the world. The only cure for preeclampsia is early delivery of the baby and the placenta. So tremendous strides need to be made to diagnose it early and reduce the severity of symptoms, and hopefully in the future, find new treatments. Our client, myself, and the rest of my group are all interested in women's health, so this project was a wonderful match. Now I will briefly introduce you to trophoblasts. In early pregnancy, trophoblast cells, excuse me, which come from the early embryo, migrate into uterine tissue and sort of invade the uterine arteries in a layer called the decidua. And when they invade these arteries, they cause a phenomenon called uterine remodeling. In normal remodeling conditions, trophoblasts are able to increase the diameter of the arteries and properly regulate the exchange of maternal fetal circulation. However, in preeclampsia, this invasion and remodeling is insufficient. So essentially, trophoblasts do not invade far enough, vessels don't get wide enough, the baby does not get enough circulation, and mom develops high blood pressure. While it is known that preeclampsia is caused by inadequate trophoblast invasion, what causes this invasion itself is understudied because existing models of the phenomena are limited. Oops. So briefly, there are three main categories of models which have been used to study trophoblast invasion. The first is animal models, but there are methodological and ethical restrictions which make these difficult to obtain and use. And if you do manage to obtain an explant from an animal, they have poor viability, meaning they don't really survive outside of the body for long. Further, no animals beyond humans actually experience preeclampsia. Therefore, no animal model has physiological proximity to humans. Next, in vitro models are a great way to get around the aforementioned limitations. Essentially, these consist of glass or plastic compartments and dishes with cell cultures in them that are used to study certain phenomena outside of an organism. But these too have limited physiological validity, and tests capture only a few parts of the environment in vivo or in the body of the organism. And finally, organ-on-a-chips in general do an incredible job of mimicking the in vivo physiological environments, and they've been largely successful in other clinical contexts. However, existing uterine models still lack the ability to fully recapitulate the microenvironment of the uterus, and they're missing key microenvironmental factors, or as we call them, cues. So we decided to pursue a novel organ-on-a-chip model for our device due to the many advantages that I just mentioned. So what exactly is an organ-on-a-chip? At the start of this process, I had no idea either. An organ-on-a-chip is a microfluidic device, meaning it's a small plastic chip with microchannels in it to allow for the flow of cells, nutrients, and other fluids for experimentation. These devices are structured to mimic the topography and chemical environment of different organs. So on the left is a real image of one of these chips. As you can see, the channels within the chips can be configured into different ways to represent different organs, like the ones on the right, such as the heart, liver, brain, and intestines. And these structures essentially make it so that this tiny device performs functions in a way that's tremendously similar to the organ itself, just on a really small scale. Our client actually has an incredible organ-on-a-chip platform called the Predict 96, which captures the physiology of several key organs, as you can see on the slide. Though the Draper platform can be used for many tissue types, it's not equipped for the specific question of trophoblast invasion in response to different cues. So that's where we came in with a novel design. Now back to our actual project, I will contextualize our deliverables and our objectives. The first objective was to develop a novel version of these organ-on-a-chips for the uterus. We were all chosen by our client, Draper, to develop a way to study trophoblast invasion. The intended deliverable was essentially a compilation of protocols, designs, and dimensions which we could deliver to our client for their own use in their lab. On a larger scale, we hoped that this device would contribute to the understanding of the pathological state of uterine tissue, or as you recall, what makes trophoblast invasion inadequate. Ultimately, we see better models as the way forward in treating preeclampsia, leading to the identification of treatment targets and the establishment of better diagnostic markers. Now here's what we actually did. First, we conducted tons of literature reviews on the microenvironment of the uterus, what current models had to offer, and how organ-on-a-chips even worked. And furthermore, we actually had to look up how to even mimic tissue on a small scale. The outcome of this phase was that we determined tissue stiffness, so essentially the mechanical environment of the uterus, to be lacking in most models and yet to have a significant role in trophoblast behavior. We now also had the technological basis to design a device like this. Next, we started to actually plan experiments we would perform with a device. For example, we 3D printed a mold that would be used to make a simplified version of a device and planned protocols to generate a stiffness gradient within a hydrogel, which is just a biologically sensitive jello, and that would mimic our tissue. However, our client informed us we'd be unable to access the labs to conduct these experiments due to COVID, so we moved right into the theoretical design of a uterus on a chip, and I'm about to show you what we came up with. So here's our uterus on a chip paralleled with the actual human uterine tissue. The idea is that the trophoblast cells in blue will travel through the chip in the same way they would in the body. So they'll start in the first compartment, or the chip's placenta, and then they will move into compartment 2, our decidua, and then into our myometrium, compartment 3. Each compartment has physiologically relevant properties pertaining to each layer of tissue in vivo. These are the renderings I developed for our device using Creo, which is a computer-aided design software that can later be used to manufacture the device. You'll notice our device has 3 separable compartments, and the idea is that in the future, researchers can actually rearrange compartments or even add more to increase the testing variables. And just a brief comment on our dimensions, you can see on the left just how small the device is. Dimensions were chosen very carefully based on the environment in vivo, and this technique is consistent with how organ-on-a-chip models have topographical similarity to environments in the body. And I'll now walk you through a schematic detailing the function of our device and how it should be used as a testing platform for trophoblast invasion. So the inlets on the left essentially provide additional modularity for researchers to input microenvironmental cues beyond just stiffness, such as oxygen or ethanol, because those have been found to sort of impact trophoblast behavior as well. Trophoblasts will then be seated into a gel in the compartment labeled 1, and prior to their movement into the next compartment, they can then be selectively saturated with these cues, which will be passively mixed in the zigzag microfluidic channel you see in gray. The trophoblasts will probe their environment and move into the adjacent gel in compartment 2, which has a range of stiffnesses meant to capture a large range of decidual stiffnesses, which are either healthy or pathological. Finally, the cells which do continue to migrate through the gel, depending on where they are in this gradient, will reach the myometrial stiffness hydrogel in compartment 3. This has a much higher stiffness than the decidua, and trophoblasts are expected to stop migrating here in vivo. The entire experiment will be performed under a device called a confocal microscope, and essentially this gives you data over time. The imaging obtained from this will allow for the determination of both the trophoblast cell velocities and the distance that they travel through the device, and hence how stiffness is affecting their behavior. So now, with our device designed, our methods outlined, and literature reviews conducted, we are passing on our work to our client Draper. In conclusion, by designing this uterus on a chip, we've provided researchers and our clients specifically with a novel, modular, and adaptable device to study trophoblasts. Next steps will be to actually run experiments we outlined, fabricate the device using traditional microfluidic techniques. And in terms of clinical applications, as I discussed previously, we hope that the better understanding of trophoblasts and the environmental cues which affect them will lead to better diagnostic markers for preeclampsia. Finally, it's been such a treat to work on this project from the vantage point of women's health. Tackling an issue that accounts for 50,000 plus maternal deaths per year and innumerable complications has been especially meaningful to me as a young female biomedical engineer. I'd like to thank our client Draper, BU's College of Engineering, Dr. Yasin Abbas, who gave his time to discuss his thesis about trophoblasts and inform our research, and finally the Honors College for their support through this journey. Thanks for your time, and I will now take questions.